Okay, so glyceraldehyde 3-phosphate dehydrogenase reactions are going to be coupled. The reaction is going to be linked by the formation of an energy-rich thioester intermediate in the active site of the GAPDH. This will preserve much of the free energy released in the oxidation reaction. So this would be your free energy versus reaction progress if you were just to look at the enzyme reactants and enzyme products. And what we can see here is that we would have our oxidation occur, and then our next stable intermediate here would be an extremely large delta G. Okay, so we drop right down into here, and then your acetylphosphate formation to get to our enzyme products would have to have this huge activation energy to get up here, right? What does that mean when you have a huge activation energy to overcome? It's very slow, right? Or yes, the number of collisions that will overcome that is much less likely, right? They mean the same thing ultimately. So here we have that free energy versus reaction progress, and what we can see is that this is where we form that thioester intermediate instead. And so we have our enzyme reactants, they oxidate, and then the thioester keeps that as a higher energy form. If we didn't have the thioester forming, we would have a much more efficient intermediate. It'd be much more thermodynamically favored, but when we try to do the second step, it would be so slow that we would never get it done, right? So again, it's that use of a intermediate values. Does that make sense? Okay. Kind of like a roller coaster. They don't just bottom you out in the first go, do they? No, they go like half of what they could do, and then they bring you back up and do some more. Okay? Because if you had just bottomed you out the first time, you'd run out of energy right away. Yeah, you won't be able to go up the next ones, right? Okay. And so again, this is going to be the much more efficient kinetically, not thermodynamically. This would actually be favorable thermodynamically for this step. Okay. Note though, how does that affect your overall kinetics? Does that affect your overall reactions kinetics? No, because they're state functions, right? So all that matters is enzyme reactants, enzyme products for the overall kinetics. Or the overall, excuse me, not kinetics. The overall thermodynamics. Okay? The kinetics, it makes a huge difference. Your active site of your GAPDH, this can include a reactive cis residue. That's where you get your thio. NAD+, and a crucial his. Step 1 is going to be the aldehyde substrate reacts with the sulfhydryl group of the cis-149 on the enzyme. This is going to form a hemi-thioacetyl. Do you guys remember your hemiacetyl? Okay. It was a Gen Chem 1 biochemistry. We, at the beginning, we had talked about the hemiacetyls or cicetyls. Okay. Yeah, instead of being the oxygen, though, remember it's a sulfur instead of oxygen. Step 2, this is the hydride ion is transferred to NAD+, bound to the enzyme to yield NADH and a thioester intermediate. This is favored by the deprotonation of the hemi-thioacetyl by his 1676. Oh, the juniper berry tea is not working well in my throat. Step 3, NADH leaves and is replaced by NAD+. Probably because I didn't have water while I was drinking. It's too concentrated. The NAD+ is going to polarize the thioester intermediate to facilitate the attack by the orthophosphate. Step 4 is going to be the orthophosphate will attack the thioester to form the 1,3-BPG and free the cysteine residue. This right here is that active site. And so right here, we actually see the NAD+. Can everybody see it okay? Okay. And this is that adenosine head, phosphate portion right here. Okay. Everybody good with that? Fantastic. Okay, what's that? Looks awfully like a sugar, doesn't it? It's a sugar. Ribose, right? Okay, so this right here is that cis-149 residue. This is the his-176 residue. This is how it actually fits together. Everybody can see that okay? So that's the NAD plus, cis, his. Okay. And that's that passover that we looked at, where it's going to have this act as sort of the thioester intermediate portion. And this right here is just a really good group to put on and off protons, right? Because it has that resonance stabilization. Okay, so this right here is going to be your catalytic mechanism. So again, remember this right here is going to be the glyceraldehyde, 3-phosphate. Here we have the NAD+, that's that coenzyme we need, right? It's going to be part of this reaction. We need to transfer the electron density to that, okay? Remember, that's our electron transporter, isn't it? Okay. And so what's going to happen here is the first thing we're going to do is we're going to make a thioester linkage. Can everybody see it okay? Okay. And that's that right here. Notice that this is just straight up that same sort of reaction you would have seen back when we did like the SN2s and stuff, right? Where we have that sulfur making a bond with a active carbon, right? That carbonyl. Okay, so what's going to then happen is we're going to get the intermediate. This is going to be the hemethioacetal. So this one right here is we have the thiol, we have the alcohol here. This is going to have the proton from this right here. Okay. Now what's going to happen is we now have this next to the NAD+. It's also next to the his residue. The his residue is a very nice group for taking on and off protons, right? It's going to weaken those connections. And so the alcohol, which is not normally super acidic, right, is going to give off its proton into a bond with this. Okay? This is going to allow these electrons up here to come down into the carbonyl bond here. And that's your thioester, by the way. That's the actual thioester intermediate. Okay. This at this point is the hemethioacetal. Hemi meaning you're making one of them is attached, thiol, the thiol's attached, instead of being a thiol, which is the alcohol version. It's a thioester in this case, or a thioacetal. And then this right here is the acetyl portion right there. Hemi meaning half, thioacetal. Okay, this is transferring the H over here. That's how it got that extra electron density to make that bond. There. Does anybody have any questions about that step so far? Okay. At this point, what have we transferred? We have transferred a hydrogen, and we have transferred what with this hydrogen? Transferred electrons, haven't we? Notice it went from a positive to a neutral. Okay. Okay. And so that's actually the hydride shift. That's the moving of a hydride, a proton with electron density on it, right? Okay. Okay, so this right here again is positive charge. That's the one that's taking that positive and stabilizing it at this point. And this right here is the thioester. Note that thioester is making this less stable than it would normally be. It's a more reactive one than it would normally be for this reaction. Okay? And so what's going to happen next is your NAD plus is going to go to NADH, and we're going to swap these out. So your NADH here is going to detach. We're going to get a new NAD plus in. Oh, I just realized that arrow is the wrong way. Shit. Oh, that's interesting. Your book normally does not make mistakes. Like, it's a very good book. But did you guys catch that? Are you swapping an NAD plus green for an NADH blue and red? No, you're going the reverse direction there, aren't you? Ah, shit. That's not a huge mistake, but yeah, It's very rare to catch your book in a mistake. It's a really good book. Shit. Anyways, we have our thioester intermediate. We've swapped out the NADH, which is carrying electron density now, went to the NAD+. And so the next part here is going to be taking an orthophosphate, and we're going to form that phosphorylation. That will give us our product. Notice that does break the thioester, and that does create it as a more energetically favorable reaction than it would normally be. Okay? And you can see that back up here if you look closely. So normally it would be a huge energetically unfavorable reaction, right? Even discounting the entire kinetics issue. Okay? Here, though, it's actually energetically favorable to take on that phosphorylation. Okay? And that's why we have that nice intermediate there, even though it's higher energy than it normally would. Okay. Okay. Does anybody have any questions about that? Okay. Anybody get confused? No? Fantastic. See, it didn't matter that they switched the arrow. It's so rare. I don't know if I've ever caught a mistake in this book before. So ATP is going to be formed by the phosphoryl transfer from 1,3-biphosphoglycerate. 1,3-BPG has greater phosphoryl transfer potential than the ATP. This can be used to power the ATP synthesis. Actually, I would be curious, by the way, if anybody wants to check in their book and see if they updated the figure without updating my copies, which I downloaded at the beginning of the semester and put in. Because I don't know if I'll get around to that, but it would be interesting. Hopefully they did, and they just left me behind. That sounds about right. Yeah, it should be at the more frontish. It's this, if you just look through the chapter for this figure, it'll be easiest probably. Okay. Now, notice, by the way, that the energy for this carbon oxidation here that we've performed is actually being converted here at this final step into that high phosphoryl transfer potential. So what we're doing is we're taking that energy for the oxidation and we're putting that towards a phosphoryl transfer, setting up a phosphoryl transfer, which is that sliding scale of which is more higher energy and lower energy, right? Yeah. They fixed it? They haven't? Oh, that's interesting. It's really rare. By the way, if you want to, sometimes they give gift cards for finding mistakes in these books. So if you want to, you can report it to the book company, and they'll put it on their docket to fix it. And they oftentimes do give like little gift cards and stuff, or discounts, or. Yeah. So 1,3-BPG has greater phosphoryl transfer potential than ATP. This can be used to power your ATP synthesis. The phosphoglycerate kinase catalyzes the transfer of the phosphoryl group from 1,3-BPG to ADP. This is going to yield ATP and 3-phosphoryl glycerate. ATP is going to be formed by substrate level phosphorylation, and this is actually what we see right here. And so we have the 1,3-bisphosphorylglycerate. We're reacting with ADP and the presence of an H+. And the phosphoglycerate kinase, notice again that name, phosphoglycerate, this one right here, kinase. It's transferring a phosphate off of, well, it'd be transferring it onto this group if you're going this direction, right? Which is kinase. But if we look this direction, it transfers it off of the thing that's not the substrate. Okay? Okay. Anybody have any questions about that? All right, so additional ATP is going to be generated with the formation of the pyruvate. The phosphoglycerate mutase is going to catalyze the conversion of 3-phosphoglycerate, right there, to the 2-phosphoglycerate. This will require catalytic amounts of 2,3-BPG to maintain an active site his residue in a phosphorylated form. And so right here is that reaction. Step 1 of this is phosphoryl group is transferred to the 3-phosphoglycerate. This forms 2,3-BPG. So here's our enzyme with the his phosphate on it. This is the 3-phosphoglycerate right here. This is then going to transfer that phosphate over to the 3-phosphoglycerate. This will make our 2,3-BPG, which is going to be that intermediate in this reaction. Okay? I know you guys were shocked, right? Isn't that interesting that biochemistry, we take organic chemistry reactions and we just put them down, collapse them down? Yeah. Yeah. Typically, these ones that you're seeing, by the way, are biologically important in some way. So the 2,3-BPG is then going to be converted to the 2-phosphoglycerate. The mutase will retain the phosphoryl group to regenerate the modified histidine. This is, again, your enzyme. It has the histidine residue right here, so it can have that out there. So that's what's going to be bonding the phosphate. We have our 2,3-biphosphoglycerate, excuse me. This is then going to be taking that phosphate from that group. This will leave us with the 2-phospho left on there. Notice that because of that, we now went from 3 to 2. What a weird way to do it, right? Instead of moving it, we're literally swapping them on and off. See that? So where does the phospho that's on here come from? The previous 3-phosphoglycerate, right? Not the current one. Okay? Okay, so again, that's the net reaction, which you guys have been seeing in this overall flow of reactions. Okay? The enolase is going to catalyze the dehydration of the 2-phosphoglycerate to the phosphoenol pyruvate. Phosphoenol pyruvate is incredibly common, and if you go into like a biochemistry biology field or a medical school, you're going to see PEP a lot, okay, PEP. PEP has a high phosphoryl transfer potential because the phosphoryl group is going to trap the molecule on the unstable enol form here. So remember how our enol forms are extremely unstable typically? Well, this one actually traps that in that form, okay? And what that means is we've actually trapped it in extremely high energy form, and we set it up to react fairly easily to form anything else, okay? And so, again, this is the 2-phosphoglycerate that we just talked about. This is the enolase. It's going to form what again? An enol form. And we're going to trap it in the enol form because of that phosphate, aren't we? Okay. And again, it does it by removing water, which is a dehydration reaction. So we remove water across where our double bond is going to form, right here. And we form a double bond. Okay. And remember again, this is that enol form that we have trapped it in. We have the phosphate there, like a protecting group, right? And it's kind of the same thing, actually. It's the biological version of that, where we're protecting it to keep it in the high energy enol form. And then we can put in another reaction, which will intermediate removing that phosphate to do other reactions, right? Very high potential energy. So the pyruvate kinase is going to catalyze the transfer of a phosphoryl group from PEP to ADP. So we put it up on the hill, and what do? We knock the boulder off so it can smash down and generate some energy for us, right? Okay. Or another way of saying that is we built a dam, right? We put a big floodplain behind it so we can increase the pressure. Okay? Okay. And so again, the pyruvate, the phosphoenol pyruvate here, very high energy, very high phosphoryl transfer potential. We're going to have ADP. Remember, in almost all these reactions, we end up having an H plus as well. Okay? It's very, very common. This is then going to react by the enzyme pyruvate kinase, excuse me. It's funny. Thursday I actually do pretty good, but Friday is the day that I usually end up having issues, right? Which is, it's like my body delays the reaction. So remember again the naming here. The kinases, they do what? Add a phosphate. So if we go the reverse direction, we remove the phosphate. And that means the substrate's always the one that's at the end, if we're taking a phosphate and putting it on ATP, right? Okay. Otherwise, these would all have to be called ADP kinase. That wouldn't make any damn sense, right? Yeah. How would you like 100 different ADP kinases? If you remember. Yeah, that'd be stupid. Okay. So this right here is the pyruvate. Now remember that because once we chop that pyruvate off, we're actually going to be extremely favorable to get rid of the enol form, right? Enols are not stable, okay? And in the presence of any sort of acid source, like water, they're going to collapse down into, in this case, pyruvate, which is the carbonyl, right? Anybody have any questions about that? Okay, fantastic. Very good. Okay, by the way, this pyruvate does have the enol form. This is going to be going back and forth in solution. It just heavily favors the pyruvate. Okay, so the vast majority is going to be in the pyruvate form. And this is so much energy collapsing down this direction that it'll basically go to completion. Okay, you have a lot of energy driving it. These are the final three reactions in glycolysis. Remember, that's the chopping up the glucose that we started this with, right? I know it's been so long, it feels like we aren't even on glucose anymore, doesn't it? But we still are. Okay, so again, we went from the 3-phosphoglycerate. That was phosphoglycerate mutase. This gave us 2-phosphoglycerate. That reaction did not work out the way you thought it would, right? Was anybody expecting us to just replace the damn phosphate with a different phosphate? Here, this tire and this tire don't look quite right. Let's just bring in 2 new tires. We won't rotate them or anything. And so this right there, this is going to be the enolase. This is that set it up to be high energy and knock it down again. Okay? And so see the same patterns we see over and over? Set it up, knock it down. Set it up, rearrange, knock it down. Okay? So basically the 2 patterns. And then we end this on pyruvate. Now again, our net reaction in glycolysis is going to be glucose plus 2 orthophosphates plus 2 ADPs plus 2 NAD pluses. This yields 2 pyruvates, 2 ATP, 2 NADH, 2 H pluses, and 2 waters. Okay? That would be your overall reaction for that entire pathway. The energy released in glycolysis is going to be about negative 90 kilojoules per mole. And so that's your thermodynamic driving force for this overall reaction. Okay? Now remember, does this thermodynamic driving force, does this directly affect each of those equilibriums? No, we actually have some steps in there that the equilibrium favors the other direction, doesn't it? Yeah, we couple them with some other stuff to help it drive forward sometimes. But every now and then, we're not clearly favoring one side, right? We have equilibriums. There's a lot of those steps. Now, there is a caveat though. Indirectly, what happens? If you have a thermodynamic that favors one side, what that means is that you're always going to have a lack of one side in terms of reactants versus products, right? And so if we're continuously removing stuff from one side of an equilibrium, what does that do to it? Drives it forward, doesn't it? Yeah. And so when you have an overall thermodynamic, it does actually feed through the entire pathway, okay? It just doesn't do it individual energetic steps, right? It does it by using up the products so you then drive the equilibrium, okay? Okay, this right here is your reactions of glycolysis. Listed all together like this, it's a bit of a thing, isn't it? Yeah. Okay. Was that as much energy being released as you thought it was going to be? No, it isn't actually that much energy being released, is it? Yet it's strongly driven forward. I mean, that's a pretty good number. But you would have thought it would be a lot more, wouldn't you? Considering like general chemistry, organic chemistry, we're in the 200, 300s all the time. This right here is your individual delta G's. And what does that mean, by the way? Which direction is that spontaneous in? The reverse. That's why I was mentioning that, where it's what's driving these things forwards is the fact that we're using up the products in the next steps. Okay? So literally these steps right here are basically at equilibrium, right? They have very, very small delta G's. And then this step right here is using up this one's product when making it is using up this one's product, etc., right? Those are pushing those equilibriums through. Okay? Okay. And vice versa. This making product is building up reactant. And so it's helping to push these equilibriums. Okay? Okay, so the NAD+, this is going to be derived from vitamin niacin. This is B3. The GAPDH reduces the NAD+ to NADH. The NAD+ must be regenerated for glycolysis to proceed. If you run out of NAD+, what's going to happen? You run out of one of the substrates, don't you? Those are one of the things that has to be bound to the enzyme for this reaction to occur. And so it will halt at those steps. NAD+, this can be regenerated a number of different ways, actually, but these are the big ones that we need to concentrate for this, what we're doing, okay? That metabolic pathways we're looking at. The oxidation of pyruvate to CO2 in the presence of O2, that's one of the big ones. The fermentation of pyruvate to ethanol or lactate in the absence of O2. Ethanol being the one that you get for like the fermenting bacterias and the fermenting yeast and stuff. You know, the beer. I guess. Does whiskey or beer make more sense down here? Equalish? Okay. Okay. I mean, technically the whiskeys are more famous from down here, but yeah. Like if you ask anybody else about Tennessee other than Tennessee, it's whiskey. Yeah. Okay, anyways, us, we do the lactate, right? Okay. Otherwise, do you know what would happen if we had the fermentation pathway? When you exercise, you'd get drunk. Now, admittedly, you actually do have euphoria sense when you exercise because of certain pathways build up and cause you to actually start pumping euphoria. Yeah. The drugs that would be responsible for euphoria in your body. But anyways, obviously not fermentation at us. The exercise would be so much more interesting. Yeah, could you imagine the sports? Like endurance running? Be a whole different event. Fermentation, this is an ATP generating process in which organic compounds act as both electron donors and acceptors. Only in my class. So, pyruvate has many possible fates. Pyruvate is one of these ubiquitous molecules again. We'll actually see pyruvate in other pathways later on, and they're all linked, okay? Though they can be contained from each other sometimes. So right here, this is the pyruvate going to acetyl-CoA. This is actually going to lose CO2. Then you go to acetyl-CoA. Then you'll go in for further oxidation. This is the citric acid cycle oxidative phosphorylation pathway. Okay? This is in animals like us, or any organism like us, the aerobic organisms. This is the vastly higher energy pathway. Okay? Way, way higher energy. And you can very easily tell that because of the CO2s. Okay? Because we're completing the oxidations. This right here is the pyruvate going to lactate. This is going to use NADH to start this off, going to NAD+. What does that tell you about this pathway? This is not efficient. And you actually have to use up NADH just to start the damn thing. NADH is energy. It's oxidation reduction energy, isn't it? So this is not efficient. It's incredibly inefficient. Okay? It's a really shit pathway. Only happens because we literally just can't do anything else. This right here is the pyruvate going to acetaldehyde. This actually does require a CO2 coming off at the beginning. Anybody ever heard of carbonated beer? And carbonated, like sparkling wines or champagnes? Okay. Did you know that only one region can actually make champagne? Champagne, France. All the other ones are actually sparkling wines. They're not champagnes. Yeah, it's bullshit advertising. Anyways, that's why you get carbonation in those. That's why the old like ginger ales, ginger beers, those used to have alcohol, is because that used to be the original carbonation method. And then we invented pressurization due to, having machines to do it and concentrating the CO2 on stuff, and we were able to carbonate stuff otherwise. But yeah, this is the carbonation right there, in case you're ever wondering. This then is going to give you acetaldehyde. It's going to use up NADH, going to NAD plus to give you the ethanol. This will overall, for both of these pathways, produce energy. This one tends to produce a bit more. You can actually kind of tell that because you're making CO2 in here. But anybody know what the bad part about this is? What are acetaldehyde and ethanol? They're products, but they're also poisons. Yeah, do you know what happens if you let just the yeast in beer just keep producing ethanol as much as it wants to? Do you get beer and it has live yeast in it? No. No, it kills itself. Okay? It actually kills itself. There's a limit to how much alcohol it can produce. That's why beers have pretty hard limits of alcohol, because they don't do distillations on them. To get any higher, you have to distill it. Okay? And that's actually what's happening here, and that's why we don't use this, is because these two things are very small molecules. They have a tendency to go in cells easily. They're very hard to get rid of very quickly. You'd have to continuously basically urinate them out. So if we were to use fermentation, we'd be running and pissing at the same time. It would be really disgusting. It would not work well, let's put it that way. Or else we would have acetaldehyde and ethanol spreading all throughout our body and causing havoc, wouldn't we? Okay? On the other hand, lactate can actually be concentrated and removed because it can't easily pass membranes. Okay? That's why that pathway diverges, is because when we're working our muscles, we would build up ethanol all throughout our body, and we'd have a huge time trying to isolate it. It wouldn't be an issue. We wouldn't be able to. Yeah. Okay. Anybody have any questions about that? You didn't know that, did you? Yeah. Kind of an interesting side note. Yeah. Sorry if I wasted a bunch of time on that. The ethanol fermentation. So this is going to be the conversion of glucose into ethanol. It's going to happen in anaerobic conditions, though do note you need to have some degree of gas exchange because the CO2 will actually diffuse itself, but when you're in an immediate environment, it can have issues diffusing itself. Okay? That's why we get build up of CO2 in the solution, and that gives carbonation. If you carbonated your blood, by the way, would that be good? That would also be a problematic, right? You'd end up with carbonic acid and you'd acidify your blood even worse than you do with the lactic acid. It typically occurs in things like yeast, though it can occur in other microorganisms. There's actually bacteria that will also do fermentation. And there's, I believe there's actually fungi as well. So anyways, yeast is being the big one that we know. This is used for brewing and winemaking. It can also happen naturally. Some berries end up getting a buildup of fermentation in them. You can get the monkeys and the and the birds drunk. Then they fly into things. Yeah, which is not as funny, but. So step 1, pyruvate decarboxylase catalyzes the decarboxylation of pyruvate. This is going to require a coenzyme thiamine pyrophosphate. This is derived from vitamin thiamine B1. Step 2 is going to be the alcohol dehydrogenase catalyzes the reduction of the acetaldehyde to ethanol by NADH. Do note that this is the step it's actually very important. I know I talked about it as if it was a bad thing, but it's a bad thing from energetics. It's a very good thing for being able to continue glycolysis, okay? Because we don't want to have to make a new NAD plus every time, right? And so this is what's giving us back our NAD plus to feed back into glycolysis and continue that reaction pathway while we're doing the rest of this, okay? Okay, and so this right here is going to be where pyruvate is having the pyruvate decarboxylase going to acetaldehyde. This is where we use an H plus to get a CO2. And notice that this is the CO2 right here. That's that carboxylate group right there. Notice the name of this. Pyruvate, substrate, decarboxylase. We're removing the carboxylate. Yeah, in CO2, right? Yeah. So literally decarboxylating it. An ACE because it's an enzyme, right? This is going to give us the acetaldehyde. By the way, yes, this is actually poisonous to humans. And this is actually basically the reverse reaction we do in our body right here. The second part, in case you're wondering. Okay? So this right here is the acetaldehyde here. And this is going to then use NADH plus an H plus. By the way, remember what I was saying, that these are usually called NADH2 plus? That's why. Is almost always comes with an H plus. I can't think of any situation where it doesn't. Okay? This gives us NAD plus. It regenerates that NAD plus we need in the glycolysis. This is the alcohol dehydrogenase. Notice that this is actually reversed, isn't it? This alcohol dehydrogenase, the alcohol is over here, ethanol. Everybody knows ethanol is the alcohol we drink, right? Okay, I pointed that out enough times. Fantastic. I'm glad I'm having an effect on you. Okay, and so this is the alcohol that we will dehydrogenate. Dehydrogenate meaning removing hydrogens from, right? See that? We removed hydrogen across that oxygen-carbon bond, forming the carbonyl. Okay. Anybody know why that would be reversed in this one? It's the same enzyme as another enzyme that we would have looked at in humans, and it would have been really important to humans because we drink alcohol. And in the case of us drinking the alcohol, we don't make it, we consume it. Yeah. So this enzyme's the same sort of one that we would see in a human. It's very similar. And obviously the reaction at that point would have been the reverse reaction, right? And so named off of that. Okay? Okay, so the active site for the alcohol dehydrogenase, this right here is the active site. This is the hydride acceptor right here, which is going to be the acetaldehyde. This is the hydride donor right here, which is the NADH. Notice, what does hydride donor, hydride acceptor mean? What are you transferring? Anybody remember what your hydride is? Chloride is the negative ion of chlorine, right? Ion of hydrogen, exactly. Yeah, you got it. So hydrogen neutral, hydride negative. Okay? And then typically we just call it proton if it's positive, right? Yeah. Or we may go fancy and be hydronium because we reacted with water. Yeah. But there you go. So this active site here, it actually does contain a zinc ion. This is coordinated to the sulfurs of 2 cis residues and a nitrogen atom of the his. This is the his down here, by the way. It's hard to see, but it's that third one on the bottom. Can everybody see it okay? Okay, so the zinc here, this is actually going to be what polarizes your carbonyl group of your substrate. This is going to favor that transfer of that hydride because the carbonyl is now pulling more electron density away from the carbon, right? Okay. Everybody see it okay? Making sense? Not beautiful. So, by the way, if you ever have trouble with your ability to process ethanol, do you know what most of those supplements have in them? If you were going to put a metal to make this work better, which would it be? Trace metal that people might not get enough in their diet. What's right there? Zinc. Yeah. So you need zinc for the enzyme to break down the alcohol, right? If you don't get zinc, guess what? Zinc actually has a bunch of properties. You can look it up sometime. It's interesting. Do you know what the most effective treatment that was done for COVID was? Zinc supplements. Magnesium and zinc supplements. They were the most effective treatment for COVID during COVID era. Yeah. They were actually more effective than the shots. Whoa, huh? Yeah, it's pretty crazy shit. COVID was weird. People have done some really good research since then, like during COVID. Really bad research, just fucking awful. But like 2 years after COVID, really good research, really interesting shit. Look it up sometime. Pretty cool stuff. So the net reaction in this anaerobic process would be this reaction right here. We have glucose plus 2 orthophosphates plus 2 ADPs plus 2 H pluses. This is going to give us 2 ethanol, 2 CO2s, and 2 ATPs, and 2 waters. The NADH generated by the oxidation of GAP is actually going to be consumed in the reduction of the acetaldehyde to ethanol. And so notice again that NADH that we were talking about before, we have consumed that, okay? And why is that important? Well, what did we need that NAD plus to go back and do? Did we just do glycolysis once? We're good. Now we can live 100 years? Yeah, we need to keep doing that, right? That pathway needs to keep feeding through. We're going to continuously need that energy and the materials being made from it, okay? Okay, and so right here, this is the NADH being produced in the glycolysis. We're going to be having it reoxidized to form the NAD+, and then it's going to come back into glycolysis and continue on and on and on and on. And you keep on that treadmill until you die. Okay. So I know the work treadmill, it's real, and it's biologically enforced, okay? So this is the true work treadmill, right here, at least for yeast. We'll get into us in a bit. This is the last lactic acid fermentation. So your lactic acid fermentation is the conversion of glucose into lactate. This is, again, in anaerobic conditions. This occurs in microorganisms and animal tissues, including skeletal muscles. This is our version of the one, right? Because we can't do the ethanol one, okay? Because we'd poison ourselves doing it. And so that anything that has a lot of, basically anything that has a lot of cells that would be next to each other tends to move away from the alcohol version, what we typically call fermentation, okay? By the way, they both are fermentation. Did you catch that? Yeah. We're just doing, it's a fermentation. It's mostly the same process. We're just not doing ethanol, okay? And so the ethanol one gets called fermentation. This one typically does not, but they are fermentations. Yeah. The lactase dehydrogenase is actually going to catalyze the reduction of pyruvate by NADH to lactase. And so again, we're going to go from pyruvate, in this case, we're going to lactate. And this is going NADH to NAD+. This is providing that hydrogen, and it's directly doing that reduction right here, okay? Again, that's lactate dehydrogenase because the lactate here is being dehydrogenating, right? It is going in the reverse direction partially because as a most of the time you see a hydrogenation, dehydrogenation, biochemistry likes to do the dehydrogenation for the enzyme name, okay? But partially it's also because the lactic acid buildup, processing it, we're going to use the same stuff, right? And so we're going to go backwards eventually. So two ATP molecules are formed in your conversion of your glucose to your lactate. The net reaction for the anaerobic process in something like an animal, where we do the lactate fermentation, is going to be glucose plus 2 orthophosphates plus 2 ADPs. This goes to lactate plus 2 ATPs plus 2 waters. The NADH generated by the oxidation of GAP is again going to be consumed, and the reduction of the pyruvate, regenerating that NAD plus to go back into our glycolysis. Okay. Now, anybody tell me, like, what the hell? We didn't really make any energy from that, did we? We make a little bit of energy, but not really anything past glucose, right? See that? How is this helping us produce more energy? We didn't actually make any additional energy, did we? That seems like a shit deal. We used up our NADH that we could have converted to ATP at some point, and we didn't get anything out of it, did we? Okay? But what did we do? We took this NADH here that is currently used up and we maybe cannot convert to ATP right away. We regenerated it, didn't we? In forms that are not going to poison us immediately. Okay? And so what we've done is we've freed up the NAD plus to come back here and help us produce those ATPs. See that? And remember that it pays us back from that initial investments. So what we've done is basically we're just harvesting those 2 ATPs from glycolysis. Okay? And we're desperate, so we're doing that. Okay? Yeah. Now, this is a little bit inefficient, but remember that we haven't actually gotten rid of our oxidation in progress. This lactate here, that has energy in it. Okay? So eventually we could go back and recycle that. Yeah. What do you notice about this reaction here? Equilibrium, right? So when we stop running for our lives, guess what's going to happen? We're going to go the reverse direction of the equilibrium, right? We'll generate NADH, and then we can start harvesting energy, and we can use the pyruvate in the citric acid cycle because we have enough oxygen. Okay? Okay. I guess we're done with time. Okay. Okay, we'll pick up here next time.